I will finish conferring with Duncan on all that and we'll send out an announcement regarding how to handle the presentations if there's a snow day. And then Friday's lecture, I will be, probably most of that will be for exam review. I did send out an announcement on points that have been added back so far. And you have your scores now on all your exams. So the final exam on Friday's lecture time, I will talk about what the final exam will cover, what the cumulative portions will be, and like at least an estimate of point value, like how much new material versus cumulative in terms of point value and overall point value. Usually the final exam is a little bit bigger than all the others, especially, well, certainly bigger than the first one, because there is a slight cumulative portion. All right. So stress, appropriately looking at stress this week for plants. And although they don't have the kind of stress you're under, they certainly are confronted with stress on a daily basis. And we've been looking at chilling and freezing, which we've all been experiencing, maybe hopefully not freezing, but when plants are undergoing this, like I was saying the other day, some are better adapted or acclimated than others. And when you look at what it does physiologically, we went through this last time, everything listed here, definitely they can measure. This last bullet point, loss of membrane function, that's really important when you think about it because membranes are supposed to be semi-fluid. And of course, membranes are not just membrane material. They have a lot of protein transporters. And so, it's a barrier. It's super important for any organism to have an intact cell membrane and a functional cell membrane. And so this loss of membrane function is often the case where if you look at the membrane, the phospholipid bilayer membrane, you have the phosphate glycerol hydrophobic or hydrophilic region, and then the fatty acid tails are the larger, in the bilayer there, that's the larger hydrophobic region. And when you look at the fatty acid tails, they can be either saturated, mono unsaturated, poly unsaturated with depending on if there's double bonds. So if there's no double bonds, we say it's saturated. And you just have single bonds between the carbons, double bond, then you start talking about, looks like the middle picture, well, yeah, the middle, no, there's a couple. So this is more than mono, it's poly because it's more than just one double bond. And so when you have the cis and trans configurations, which anytime I hear cis and trans, I kind of get triggered to organic chemistry, but not a pleasant thought. But anyways, cis and trans, you get these kind of kinks in the tails of the fatty acids where they don't line up nicely with each other anymore because they're bent and kind of kinked in different directions. And so that creates fatty acid tails next to each other that you can imagine if you start to either put pressure on them or if you put colder and colder temps where things start to gel up, it's harder for the membrane to gel if there's a lot of double bonds because all the tails are kind of kinked and bent. And so it keeps them more fluid. And so if you look at plants that are grown in areas that are, or native to areas that are go through a winter season like this, they tend to have more polyunsaturated double bonds. And so and they can do that, on the fly. And so they can switch out, so to speak, their fatty acid tails as they acclimate if they need to. And so this data here, looking at fatty acid composition, and you can see they were comparing chilling sensitive versus chilling resistant species. And you can clearly see, so here's your sensitive species here. And you can see if you look, especially if you look at the very bottom here and compare those values. So they're looking at the ratio of unsaturated to saturated. So the higher the number on that, ratio means in this case, there's more unsaturated. And so you can see the chilling resistant have higher numbers. So they have more unsaturated fatty acids. So that's something that they can do on even within their own lifetime. They can switch out their fatty acids and repair that and get it so that they're better acclimated. The other huge problem that they have to worry about, so to speak, are ice crystals. And so there've been a lot of examination of how plants deal with this, especially plants that live up in these types of weather systems where you have long extended winters. How do they not get ice crystal damage over time? And what they find is a very methodical process to, as the temperatures are dropping and you get the closer and closer to freezing temps, and then finally you dip below freezing, what tends to happen is that the ice will form first in the cell wall spaces or the intercellular spaces. So that basically you're looking at the cell wall area between neighboring cells. That's where ice will form first. So what happens is you get water moving out of the cytoplasm slowly into the cell wall space. And if ice crystals start to form, it'll be there in the cell wall space. And that's less damaging because it's just cell wall material, not precious proteins and enzymes that you see inside the cell. So as that process is happening, you see this water coming into the cell wall space. The cytoplasm starts to lose water, right? That's where it's coming from. So you're essentially dehydrating the cells to some extent. So as you have water leaving the cells, but everything else staying behind, all the proteins and enzymes and solutes and on and on and on, you're essentially creating a pretty high solute cell now. So your CISA-S value. Anyways, what this does under freezing conditions is it's almost acting like an antifreeze when you have all these solutes and not as much water in the cell. And so that's actually a good thing, believe it or not. And so they've also discovered species that are adapted to seasonal weather like this. They actually have special proteins that they will get up and running around this time of year that they actually call antifreeze proteins that will actually stop the size, the increase in size of the ice crystals. And so basically, if you have a bunch of little ice crystals, especially in the cell wall space, a bunch of little ones, that's much better than a couple of big ones because the big ones can start to puncture through the cell wall material to the cell membrane, and then you get punctured cells. So that idea of the dehydration of the cytoplasm, what you end up also seeing is you get all the leftover materials, staying there. So you get solutes, plus they will often synthesize special sugars that they essentially call antifreeze sugars that are capable of pretty much behaving like the antifreeze in your car, where it will not allow for the water to form ice crystals inside the cells. So that's the big goal for the plants. They're trying to not get ice crystal formation in the cells because they don't, of course, want the cell functions to stop, and they certainly don't want their cell membrane to get punctured. So this is just some examples of different compounds that they found in plants that are adapted to seasonal and long winters. So this process I've been describing, they refer to it as supercooling. You don't really need to know that term, but if you see that, that's pretty much what they're describing or what they're implying. They're talking about the water moving out into the cell wall space, preventing small ice or big ice crystals slowing. So basically, all of this is you factor in how well can they do that, how they have to dehydrate, but obviously not to the point of the cells dying. So that's something to consider. And are they good at keeping their ice crystal size down? And if that is all going well, and they've seen plants that can do this better than others, But basically what they see is about -40 degrees Fahrenheit is about as low as you can go temperature wise. If you go beyond that, like -42, you start to see spontaneous ice crystals that can be pretty damaging. And so you have some plants, I mean, but that's pretty amazing, -40 Fahrenheit. So that helps you to explain why some plants, they persist in such harsh environments and long winters. And you think about some of the other areas like up in the tundra, that are even more harsh than this area. You can see now, you can imagine why they're able to survive. They're just really good at all of these ideas that we're talking about. They also have been investigating hormonal control of this whole process, and they do see abscisic acid involved. And we know, abscisic acid, one of its famous abilities is to get the stomata to close down under dehydration or drought. But with this idea, what they find is that if there's essentially two ways they discovered in this experiment, you can get your plant to deal with freezing if you get them used to it. So that's the idea of acclimation, where you give them short exposure times to cold conditions, and then you get them away from that, then you do a longer and longer time. And so you just slowly get them used to the very cold conditions. So you can do that, and they did that experimentally. But the other thing they did in that experiment was they took the same plant and they sprayed it with abscisic acid and then just put it into cold conditions and looked to see what would happen. And when they did that, it greatly increased their ability to tolerate cold temps. And so either through this slow kind of methodical acclimation process or with abscisic acid, that it just allows them to deal with the freezing. And they're not quite sure exactly how this is happening. But so far they've discovered that some of those same proteins that are synthesized, those kind of antifreeze proteins, the abscisic acid seems to be involved in that process and kind of trigger it. And so far they've discovered that. The other thing that they've been playing around with, when you look at, when you're trying to figure out what this hormone does, one thing you try to do is create a mutant plant that is insensitive and then compare it. And so when they use the word insensitive, what they mean is that if they're insensitive to abscisic acid, that usually means you've messed with the receptor. And so it can't interact with abscisic acid because the receptors aren't there. And so they don't hear the signal, so to speak. They're deaf to abscisic acid. Then if you do that kind of mutant and run your experiment, they don't survive the freezing. But if you create a plant that just can't make its own abscisic acid, but it has receptors for it, and you just stopped its ability to make abscisic acid, and then you spray it with abscisic acid, you can get them to tolerate. So they're trying to tease apart exactly what's going on here, but these last two bullet points really kind of hammered home the idea that, yep, abscisic acid is necessary for part of this process of getting geared up to tolerate the freezing conditions. So I think there are some businesses down south that are using this idea, and they'll spray their crops with abscisic acid prior to hard freeze. If their plants are, they're trying to keep their plants going beyond that hard freeze. So if the hard freeze happens too early in the before harvest, there's been a couple of businesses that have tried this out, and it has helped, has worked. So they're trying to, especially in areas where it's still an active growing season, but all of a sudden they get a cold snap. That can be really challenging. They can lose massive amounts of product that way. So they're really trying to find some good solutions to this problem that aren't too expensive, or it's not going to hurt the environment in any way. So this idea that they've known about for quite a while, many decades. It's this weird phenomenon where certain bacteria that are pathogens to plants, they have this special protein that they make and then they excrete it out and it lands on the plant surface. So these bacteria are usually on leaf surfaces. They excrete this protein, so it ends up on the leaf surface. And what it does is it causes ice nucleation, which just means the protein is well designed by its chemical structure and makeup to kind of like attract ice or water and then freeze it so that you get bigger and bigger ice chunk or crystal. And you can imagine if that crystal gets big enough, you're going to start damaging plant tissues. And so from the bacterial perspective, the pathogen's perspective, this is actually a great way to get access to nutrients. You just have the ice crystal puncture the cell membranes of the plants and you suck away, so to speak, all the nutrients. So it's a pretty low cost method for the pathogen to employ, but it causes a lot of damage. This has been a problem, especially in plants that are grown in the southern states, that you get those low temps too soon in the season. So it can even happen where you look at the thermometer and it's saying that instead of it's, freezing is 32 degrees Fahrenheit, let's say it'll be like 34 degrees. And you're like, well, it's not freezing out. My plant should be fine. And yet you go and inspect your plants and they've got damage due to ice. And so what happens is they started looking at that going, what's going on? It's not cold enough for ice. What is causing this? And that's when they discovered these bacteria doing this. So they can actually create crystals under warmer conditions than what the thermometer is telling you it should be doing in terms of ice formation. So that kind of kicked off a whole bunch of research looking at how ice nucleation happens, especially with regard to when it's right around plants. And then what they tried doing, because this was a big problem, this particular pathogen was getting a nice foothold in certain crops and causing a lot of damage. So this was actually one of the cases of the first genetically modified bacteria getting put out into the environment. And so what they did is they took the pathogen, stopped its ability to make that protein, and then they sprayed the crops with that. And the idea is it should outcompete the ones that, the pathogenic ones that might be lurking there. And so they basically just kind of swamped the system with a bunch of non-pathogenic bacteria of the same species, and then they outcompeted the pathogen, and it worked. And so the problem with that was really, really early on in genetic modification, and a lot of people got really concerned that a bunch of genetically modified microbes just got sent out into the environment. And so that caused a lot of uproar and actually led to a lot of more, like, we need more regulations in place in order to do these types of experiments. So it was kind of an interesting time period in the experiments of plant pathogens. Okay, so ice formation is a problem, but many, many plants are adapted, especially native plants, they're adapted to deal with it. So the other day when I was saying that's one reason why you'd want to use seeds from plants that grew in the environment that you want to plant those seeds in, because hopefully those seeds came from adapted or acclimated plants and they have all these abilities right there ready to go. And so that's often why a lot of gardens in this area don't work so well if you use seeds from just some big seed company where they don't have acclimated seeds. So sometimes that can be a big problem. Okay, so chilling, that's all I want to say about chilling and freezing. The other, when I asked that questionnaire on what other topics, the next closest one that was several people expressed interest in was flooding. So I just want to say a few things about that to give you a taste of what plants have to deal with when they get flooded. Which with, we had an exceptionally rainy, especially fall. You certainly know that flooding can happen. And if it happens in agricultural fields, it can mean the loss of that crop entirely. Which obviously, if that's your livelihood, that's not a good idea, not a good thing. So one of the huge takeaways from this idea is that if you have plants that are not aquatic plants, that's not their native habitat, they normally lived in soil that has a lot of air spaces in the soil system, and you have a long flooding event, you basically are suffocating the roots. Roots are living tissue. They need oxygen in order to generate their ATP. And if you create flooding environments that are long-term, all the air spaces in the soil are saturated with water. Oxygen diffuses very slowly. That's the other big takeaway. Oxygen diffuses very slowly in water. So not only do you have lack of air spaces in the soil, now you have the trouble of the roots can't get enough oxygen quickly enough, and they start to die. So I'm just showing the mitochondria here because, of course, they do respiration very similar to how we do it. They have mitochondria and they're using oxygen as the terminal electron acceptor, which is this. And we didn't get into this in this class at all on how plants go through respiration. And I'm not asking you to know this. This is just a visual to get you thinking about the idea that plants have to have oxygen, just like we do. So here you see oxygen sitting at the end of this big, long electron transport chain process. And so it's sitting there as the final electron acceptor and will accept, 4 electrons, one by one by one, and then form water. So this is cooking along in your body right now, but certainly plants are doing this too. And if they don't have that oxygen, then all of this falls apart. They just can't do it. And then they end up dying because they can't end up with enough ATP to survive. So if they have this issue, that's one big problem, obviously, because they can die from lack of oxygen. But another problem complicates this. So when you have a well-drained soil, then we say it's aerobic or oxic. It has plenty, aerobic means there's oxygen. If it's a flooded soil, then especially if it's a long-term flooded soil, more than just a brief thing, then the soil can go anaerobic or anoxic without oxygen. And so if that starts to happen, you have oxygen diffusing too slowly. They don't get enough of it, and eventually they won't get any of it, and they'll end up not being able to do the respiration process. If you then when they've done timed experiments and they look at plants that aren't used to flooding, it can be just 24 hours of a flood. And that's enough to really tank the plant. They have a really hard time recovering from that and surviving it. If they take plants and experimentally that are tolerant and expose them, they have a much greater chance of surviving, and then they tend to bounce back and finish a growing season that's pretty close to what it should be. And especially, and then you got the really interesting plants, like the ones that are native to estuaries and swampland where their roots are commonly flooded. But when you look at those plants, they're adapted, where they have engineered their root tissue to deal with this lack of oxygen in particular. And I think I have some pictures here. I'll show you that in a minute. But basically what they do to those roots is that they create essentially air gaps in their tissues to allow for any oxygen that makes it in. They'll have an easier time getting that oxygen into the tissues and get it dispersed to all the cells of the root and sometimes even the base of the stem. And they will have like nice like internal routes of connection of these air spaces so that you can get a pretty efficient flow of oxygen through that area. So they give it a special name. They call it parenchyma. I would like you to know that term, please. If you're asked about flooding and how they engineer their roots, they use that term, parenchyma tissue. And so it's just really nice tissue that has lots of air gaps where you can get good oxygen diffusion. So what tends to happen is a living plant can engineer on the fly. So let's say even an adapted plant, if it'll naturally have some parenchyma tissue, but if it needs more, like, oh my goodness, this flooding event is taking a long time, we need to ramp it up a little bit, they can kick in through signal transduction, and the hormone is ethylene. Ethylene can actually like carve out some of the living cells, essentially cause apoptosis, so that you end up with more parenchyma tissue. And so it doesn't have to, it's on the fly. The living plant going through that flooding can actually get that to happen to their root system as the flooding is occurring. And so that's a very handy response. If they need to, they could kick into creating even more parenchyma tissue. So here's a image. This is root tissue. So on the left is kind of the normal process of what a root tissue looks like. And then I think you can clearly see on the right, you have all of these big gaps of, lack of cells. And so that's where you tend to see nice flow of oxygen. Once it gets into the tissue, it'll flow quite nicely and get dispersed. So and this is corn. So, corn is not an adapted plant, actually. It's not a plant that's adapted to flooding, but it is tolerant to flooding. And so on the fly, it can do this, even though really corn doesn't have parenchyma tissue, really. What's being shown here on the left is what it normally looks like, and there's not any parenchyma tissue per se. And so this is a pretty interesting example of a plant that might be slightly tolerant, but then really ramps it up and can get really nicely tolerant, at least for a stretch of time. So anaerobic, when you don't have enough oxygen in the soil and they declare it anaerobic, one of the other problems for the plant is that it creates conditions where different microbes now start to gain an advantage. And these microbes tend to be microbes that don't coexist nicely with the plant. And so most of the microbes that hang out with plants use oxygen, that coexist and help plants, they use oxygen. However, there's a bunch of microbes in the soil that prefer no oxygen. And if they start to, so if you can imagine that soil gets flooded, really, really saturated, and let's say it persists for days. Now the anaerobic microbes start to, their population sizes go up. So what's being shown here on this diagram is that some of those microbes are doing this thing off on the right here, where they're putting nitrate. So there's no oxygen to use for their version of respiration, like what you see on the left. So if they have oxygen, they'll do what's being shown on the left. They'll grab oxygen and put it as a final electron acceptor. They'll get a bunch of ATP. So what they're doing here is to get ATP, which they need to survive. But if there's no oxygen, a bunch of anaerobes will do what's shown on figure B, where they're like, okay, I don't have oxygen. I'm going to put nitrate instead and run my reactions to get my ATP that way. Well, think like a plant. If you're a plant hanging out with all these microbes that are taking nitrate and converting it to nitrite, that's not who you want as a partner. You don't want these microbes around you, all right? Because if you're a plant, you love nitrate. You want that. And now the microbes are hogging it away from you and using it. So this is actually a really big problem. If you end up with a soil system where the nitrate is being used by the microbes, plants really don't care for nitrite that much. In fact, too much of that can be not so good. They want the nitrate, NO3 minus, but the microbes are hogging it. So we're at this idea here where if the microbes are running the nitrate in place of oxygen, some microbes can do all of this in the soil. They'll take it and convert it to nitrite, which I just showed you on that previous slide. Okay, so that's what's being shown here, nitrate to nitrite. But then other microbes in the soil can push that reaction all the way into nitric oxide, nitrous oxide, dinitrogen. All of those are gases. Those go up into the atmosphere and are lost to the plant. Once it's gets out of the soil, the plant can't touch it. And the plant certainly can't touch dinitrogen. Only bacteria can take that and get it back into the soil. So this is not a good situation. So not only are the plants dealing with lack of oxygen, but now they get the wrong partners hanging out with them, doing all kinds of things that are very much against what the plant wants to do. So that's a big problem because plants need nitrogen, lots of it. So big problem. Another thing that these anaerobes are doing is they're taking a ferric, Fe3+, the ferric form of iron, and they convert it to ferrous, which in and of itself, that's not terrible. But if you have too many of them doing it, plants start to suffer because if you get too much of the Fe2+ form, it's more soluble and it's out of whack with what a plant is used to. And it actually creates toxic conditions for the plant. So it can also act, well, that should be a bullet, not a dash. The sulfate idea, this is a separate idea. So what can happen here is the sulfate gets reduced to hydrogen sulfide gas, H2S. That's a poison to any living organism. It's poisonous to us. It's poisonous to plants. Microbes, some microbes are fine with it, but most other organisms are poisoned by it. So that's a problem as well. So a bunch of anaerobes are doing that. The microbes are doing that, and that's toxic to the plant. Other microbes might be producing butyric acid, acetic acid, which enough of that building up is toxic to plants. So this is really like a double whammy. The plants are dealing with really terrible access to oxygen, plus all the stuff the microbes are doing, the anaerobic microbes are doing, is really starting to create toxicities and problems. Now, for a time, plants can do fermentation a little bit, which just means they can run physiological pathways that don't involve oxygen. But any organism that does fermentation, the payoff is pretty pathetic. You don't get a ton of ATP. And if you can imagine a mature plant getting flooded, a mature plant needs a lot of ATP on a minute by minute basis. And then all of a sudden you flood that plant. It can switch to fermentation for a while anyways, but the efficiency is terrible. So a mature plant's not going to be able to do enough in order to get its needs met. This figure is just trying to show, for those of you who are in chemistry and biochem, this may look familiar, looking at the fermentation reactions, but we're not going to get into the details of it. But if it's a visual that helps you think about fermentation, then that's why I put it there. So fermentation, especially with fully grown plants, it can help for a while, but it's not going to do much. You only get, per glucose, you only get 2 ATP molecules, so that's pretty bad. It also creates problems. All the anaerobic soil, they just have a hard time taking up minerals and they don't have enough ATP to get that to happen anyways. So it is a problem. It also creates this weird situation where if the flooding persists, you will start to get ethylene made in the plant, which is okay in the roots because it can create parenchyma tissue. So that's fine. But if this continues, sometimes what happens is you'll get ethylene migrating up through the plant body into the stem and leaves, and you'll actually get the epinasty response that you looked at with your plants in lab earlier. where they look all wilty, they droop. And so it's really weird. You'll drive by a flooded field and you'll see all these mature plants that look like they're wilting, like they're not getting water. And it's not that at all. It's an overproduction of ethylene that made it up into the shoots and is causing that epinasty. So it's kind of a weird phenomenon. This is just data that they collected from a controlled experiment where they show ACC is just the precursor to ethylene. So that's this precursor, this intermediate, and then you have ethylene. So they measured both ACC and ethylene, and they looked at flooded versus non-flooded plants. And you can see huge difference if the plants are flooded. They get this huge upswing. So it's kind of weird to see them looking like they're all wilty, but it's just the epinastic response by the plant because of too much ethylene. So when they do these experiments, they try to see, okay, if you keep the flooding going for a while, you know, they see this parenchyma tissue, they can see that plants can deal with it for a while through fermentation. But they've also found some interesting other ideas is that they'll actually see the stem biomass greatly increase. It's like the plants are trying to get above the water or at least most of their body above the water. It's kind of like treading water when you get thrown into the pool. In order to stay above the water, you try to get, in order to breathe, you try to get out of the water, right? So that's what they're trying to do. So they actually see this phenomenon where the plants get taller. And so you'll get a bunch of cell division and growth in the stem region. And it just is trying to make sure that most of their body is out of the water. And then they would have better access to oxygen. So they actually termed it an escape strategy, even though they can't run. They're not physically running and escaping. They're just trying to grow taller and escape being, fully submerged. So this is the idea of, we showed this before with gibberellic acid, being able to greatly stimulate the stems growth. And so they did some experiments here. This was, I think, rice plants that they were using, right? Yeah, rice plants. And they were doing different genetically modified rice. And so the top picture is before they started the experiment. And then you can see 16 days later, they stopped the flooding, and then they followed it for an additional 7 days. And you can see some of the plants did recover and some didn't. So the intolerant lines didn't make it, but the ones that were modified so basically it was just this idea of trying to get the plant to respond to flooding by shooting up their stem growth. So they are working on this, especially for plants that are in flooded areas, throughout the growing season, it's a risk. So they are looking at genetically modifying for flooding. Okay, but that's all I'm going to say about flooding. It's kind of just a couple of big picture ideas on that with the oxygen issue and then the microbes doing not so nice things that harm the plant in the long run. So I'm going to stop there with lecture material. And so again, with Friday, it'll be a review session. So I'll talk to you about the final exam, but come prepared to ask questions too, because we can clear up anything that needs clarification. And the final exam is next Wednesday at noon. All right, do we have any questions right now on anything? So please check your EduCat announcements before tomorrow so that you know if there is a snow day, what the plan will be, okay? Yes.
Start from the signals.
Oh, yeah, so start material will be Yep, it'll be the signals from sunlight and then moving forward, yep. And so we have signals from sunlight, signal transduction, and the hormone information in that as well. And then we got into plant secondary metabolites, plant defense mechanisms, and stress. That'll be all the new stuff. Cumulative, I still have to finish analyzing what I'm gonna put on the cumulative. I don't know yet for sure, but Friday, I will definitely tell you Friday. All right. Stay safe.